Okay, I guess we can get started. So the next speaker is Tina Ludwig, who was used to be a postdoc here in the Isotope group, and since the last few years has been running a proteomics facility associated in Munich, associated with Bernhard Küstler's lab. Tina is a very special guest because she was when we showed this timeline of all these dots, she was one of the main people who started these courses off back with SRM in 2013. So we're very happy to invite her back to tell us about some kind of general concepts in quantification and absolute quantification. Thanks, Tina.
Cool. Is it working? Yeah, because it's blinking red. I don't know if this is a bad sign. So if you don't hear me at one point, let me know. Okay, yeah, so thanks a lot, Ben, for this introduction, and I'm also very happy to be back here. And I think my lecture now will be maybe a bit different from all the other lectures that you heard so far, in a sense that it will not be very focused on DIA and SWATH. And I think it will also probably be a bit more basic. And maybe that's good because it's also so late in the evening. But what I would like to discuss a bit with you, or to look at with you together, is at some general proteomic quantification concepts which apply to no matter which type of mass spectrometry you're actually using. And I would like to focus especially then in the second part on absolute protein quantification. And whenever you have questions, really, please just stop me at any time. I'm happy to hear your questions. So I've structured my presentation in 3 parts. As I said, we will start with a very general introduction in how proteomics can be quantitative at all. And a lot of the things that I will explain there actually came up in the last 3 days already, or have been mentioned by other people already. However, I think they were not really explained maybe in detail. So hopefully you can still benefit from a little bit more basic introduction. And then in the second part, I would like to focus, as I said, on absolute protein quantification and tell you my definition about relative and absolute quantification, and look at label-based and label-free absolute quantification. And in the last part, I would just like to show you some real data as one example where we used absolute label-free abundance estimation in combination with DIA SWATH to look at Mycobacterium tuberculosis. Okay, so general introduction into the basic concepts of quantitative proteomics. So one question that you can ask right away, and probably many of you are rather new in the field, even don't know this anymore. Everybody of us does quantitative proteomics every day, but still originally in the early days of proteomics using mass spectrometry, it was thought to be a qualitative tool, right? Where people said you cannot really quantify nicely with proteomics data, you can just identify the peptides. Do you know why this was initially the thought of the field? What's the problem with quantitative mass spec data? Any ideas? Just throw them in. You're doing quantitative proteomics and you don't know why it shouldn't work. Ionization. What does it mean, ionization? Of the different peptides. Very good. Exactly. So that is also, it's not just ionization, but that's exactly what I wrote down here as well. So when we do the big problem that we see is that there is actually a rather poor correlation between the amount of a peptide that we see, between the amount of a peptide in a sample and the MS signal intensity that we see when we measure this peptide in a single spectrum. So to make this very clear, let's have a look at this example. So let's assume we are having a certain protein that we would like to analyze with mass spectrometry. We digest it into its peptides, and then each of these peptides are, of course, in theory equimolar. They have the same concentration, they come from the same starting amount of protein. However, the intensities that we will see in the mass spectrometer can be very different, by many folds different. And the reason, from a very simplified point of view, for these differences is, as you already said, ionization differences of different peptides ionize differently well in electron spray ionization. But there are also many other factors that influence these intensities that we see. The concentration is also one factor, of course, but also digestion efficiency. If the protease doesn't cleave a peptide 100% out of the mother protein, you will have different concentrations here and different intensities. Recovery of certain peptides during sample prep. very hydrophobic peptides we tend to lose in the workflow due to attachment to vessel walls or something like that. So not every peptide will make it completely to the mass spectrometer as others. The ionization effects, matrix effects, so depending on which other peptides are co-eluting at a certain time point of our gradients can influence the intensities that we see. And just the very specific properties every different sequence has. And it's the combination of all of these different factors that make up what I call now for the rest of the talk the specific MS response factor. Okay, and the problem really is that without knowing the specific response factor, we cannot just say from the intensity that we measure for a peptide what was the concentration of the protein. If we know the specific response factor, then we can draw this conclusion. Okay, so now that was of course sounds very depressing. However, what people did realize pretty fast is that if you couple a chromatography online to your mass spectrometer, and if you now measure the intensity of always the same peptide over time during chromatographic elution, and you do this with a certain speed over and over again, you can extract such a chromatographic trace. That's what we have seen already throughout the first 3 days very frequently. And what the people found out that actually either the peak height or even better the peak area of this chromatographic trace, this nicely correlates with the concentration of a peptide in the sample. So if you have a twice as high chromatographic peak, it means you have twice as much of that peptide present in the sample. And what also Alexei and I think many others already pointed out, for such a chromatographic-based quantification, it is actually super important that you can really nicely reconstruct this true chromatographic peak shape. And the most important parameter that influences this is this cycle time, as people have already told before. And that's the time that lies between the acquisition of the intensity for certain peptides. And yeah, so the rule of thumb that people work with in the field is to use 8 to 10 data points over this elution profile. We heard already people saying maybe 6 is enough, maybe 10 is better. I think 8 to 10, yeah, that's what many people try to apply. And now what do you need to do to achieve 8 to 10 data points over a chromatographic profile? Of course, this completely depends on your chromatography and on the peak width of your chromatographic trace, right? And I think many of the nano-LC systems that we work with in proteomics have maybe average peak width in the range of 30 seconds, and then a cycle time of 3 seconds is fine to give you 10 data points. If you manage to achieve a really good chromatography, which we all want, maybe only 5 seconds wide peaks, your cycle time must be also much, much faster. And that's very relevant if you do want to do quantitative proteomics for DIA SWATH, but also for shotgun proteomics, for DDA, for PRM, whatever technique you're using. Okay, so also slides like this you have already seen. Here, this is one example from me where I wanted to measure these synthetic labeled reference peptides in different concentrations and see whether peptide concentration and intensity really linearly correlate with each other. And that is one of these nice examples where this worked very well. However, and Olga mentioned this already, there is a problem with this linear dynamic range that we all assume for quantification. And the problem is it holds true for many peptides, but only for a certain range. So only for a certain intensity, only for a certain concentration range. And the upper and lower limits of this linear dynamic range is what we call the lower limit of quantification and the upper limit of quantification. And what's always happening at the sides is that we have these saturation effects. So what's happening below the lower limit of quantification is typically that we only measure background, right? Maybe we don't measure background in the fusion instruments anymore, what Ludwig showed us, but typically you just at one point measure a stable background intensity value, so that's why this flattens out, and this is what Olga also showed you before is the hockey stick picture. And at the higher end, above the upper limit of quantification, you run into problems with detector saturation typically, and then just the detector is closed and you keep on measuring the same intensities, even though the concentrations go up. Now, this is of course very logic, and but what's very important to keep in mind, and it actually holds true for any type of proteomics approach or quantification approach that you're doing, is that all of the data points that you use for quantification actually must come from this linear dynamic range. Only then the basic assumption that we have a linear dependency between intensity and concentration holds true. So if we have, for example, a case like this, that we want to compare the concentration of a peptide or a protein in two samples, and the intensity that we measure in one sample comes from this area below the linear dynamic range, the other one is happily in the range. If we now determine the ratio between these 2 points, the ratio will be erroneous, right? The ratio will be rather too small. The true value of sample 1 will probably be rather here somewhere. Okay, and this is true whether we do label-free quantification, or where these are 2 different samples that we've measured, or whether we do isotope labeled quantification, where this is a heavy and a light peptide. We always assume linearity, and therefore for a really accurate quantification, you actually should check for this range of linearity for all your measurements. It might still be possible to, from such a comparative analysis, to still find that this peptide or protein is significantly regulated. So it's not saying that you necessarily get wrong or bad results. But if your aim is to do really accurate quantification and determining the true ratio, what you will have to do is to check that all your measurements are coming from this linear range. And unfortunately, this linear range is nothing that you can just define once and then it's true forever, but it's something that is peptide, sample, and mass spectrometer dependent. Okay, and it can be different for different peptides, for different sample metrics, and for different mass spectrometer. Okay, so now let's say, yeah, that's true, and we acknowledge that, and now we want to determine the linear range. So how can you do that? What do you need to do to investigate the linear dynamic range in an experiment? Yeah? A little louder, I didn't hear you. A calibration curve. How do you do that? What does it mean? Exactly? Sorry? Of an internal standard, the illusion of an internal standard. Exactly, that's again what I have written here as well. So you would need to construct a dilution series. Typically people work with isotope labeled peptides or protein standards from which you know the exact concentration, and then you just take a representative sample matrix. So, and then you spike into this sample matrix your isotope labeled standard in different concentrations, and you measure it, and then you check, do I have this linear dynamic range? Where is my lower limit of quantification? Where is my upper limit? You can apply tools like Olga just described before to find these points in your data. However, the big problem, isotope labeled peptides or protein standards are really expensive. Pure isotope label, where you exactly know the concentration, are very expensive. So usually people do such an approach for, let's say, in the range of tens of proteins or 100 of peptides maybe, but it's just something that's too expensive to do for a lot of proteins. So let's now see what else can you do if you now want to determine linear dynamic range for thousands of proteins, for a whole proteome. Do you have any chance to do that, omitting these expensive synthetic or reference proteins? Different amount of sample. So you would just take your sample and dilute it. You could do that. Any idea what could be a problem of doing that? Just dilute a full sample and look at it. You will have a different total protein amount. In theory, the idea is great, but the total amount will change, and it will change a lot if you span a large dynamic range. And there are a lot of effects that come into play in that moment, from matrix effects, retention time differences, also ionization differences. Yeah? Super. You can try to do exactly what you described, but you correct for these differences by adding another another organism. Exactly, and that's a slide that I stole from Lindsay Pino from the McCoss group, where she proposed exactly this. So what you can do is just take one proteome and then dilute it with another proteome that should be of similar complexity. And so don't use a super complex human to dilute E. coli. That would probably not be a good idea. But you can use, for example, a labeled proteome of the same organism. So the total protein concentration in every of these tubes will be the same. And then you can determine linear correlation curves for all the peptides that you can measure, for example, by doing a DIA swath measurement of these samples. And what's different to the example that I showed you before is that you, for these type of dilution series, you no longer know the exact concentration of that peptide, which I knew before by working with the isotope label standard. You just know the total concentration that you used for that organism. So you cannot exactly say what's the concentration of this peptide, but you can still find the intensity range where this peptide behaves linearly. And that's very useful to define where does my range of linearity stop, maybe. Okay, so far we were all the time talking about chromatography-based quantification. And as I said in the beginning, chromatography-based quantification, you just need to measure always the same peptide and the intensity of the same peptide in consecutive measurements. And this can be MS1 measurements, as we do in DDA mass spectrometry, but it can also be, and also in PRM or DIA swath will provide you MS1 information, while you can also in the same way use fragment ions, MS2 intensities, and extract chromatograms. And also PRM and DIA will give you this information, and also SRM or MRM measurements. But next to this chromatogram-based quantification, there are two additional units that proteomic data possibly provide. The one additional unit that people work with for quantification is reporter ions by working with isobaric tagging like iTrack and TMT reagents. And here you do exactly what I just told you on the first slide is not a good idea to do. You compare the intensity of two different analytes in the same MS2 spectrum. However, these here are not two different peptides with TMT and iTrack labeling, but these are reporter ions. So they are thought to be exactly the same. They have the same ionization efficiency, if you like. So with these reporter ions, you can do exactly this comparison and use this for quantification. And this is then done on MS2 level only. And next to isobaric tagging and reporter ions, there's a third quantification unit that is used in the field, and that's spectrum counting. So what you do here is you simply count the MS2 spectra that have been identified for a certain protein. So these can be different peptides several times, or the same, different peptides or the same peptide several times. You simply count them all up and you use this for quantification. And these different quantification units have, of course, since a long time been compared to each other. And I think it's very clear and has been shown many times that spectral counting compared to the other two performs significantly weaker in terms of reproducibility and accuracy as well. I think spectrum counting just works reasonably well if you have really very large data sets with millions of spectra that are acquired. And I think this is also the reason why this quantification unit is definitely used less and less in the field, and yeah, hopefully probably dies out. And the other two are still equally well used very frequently in the field. Okay, so if you want to do now quantitative proteomics, what you are facing is a situation like this. There is a huge number of different approaches, methods, techniques, flavors of methods published that you can use to quantify proteins and peptides. And everybody in the field has always the same problem. You need to choose one of those for your specific project. And there are a couple of questions that define which might be the most suited method for your approach. Questions like, what type of mass spectrometer do you have access? Which type of mass spectrometric data can I acquire? How precise and accurate do your quantitative results have to be? Are you just interested in measuring proteins that are super changing or super abundant? Or do you need to measure very subtle protein changes? Then you need to maybe use another method. what type of sample are you working with? Can you do metabolic labeling with it, or can you not do it? How large is your sample number? Are we comparing 2 or 2,000 samples? How much money can you spend? How much time do you have? And also one question that is important here is, do you need relative or absolute quantitative data? And so this is the question I would like to focus on for the rest of the presentation, about the difference between these two. But maybe there are any more questions to this general introduction into how and why quantitative proteomics works or does not work. Are there any more questions? No? Yeah? Super.
I was just wondering, when you make the curves and find the aLFQ and aLFQ, the intensity or the sensitivity of the mass spectrometer is to vary over time. So how do you take that into account?
So you say when you have performed measurements like this, the sensitivity over those 8 or 10 injections did change a lot, and that you could then see different intensities there because of that?
Yeah, or if you make this curve and you decide for a span of concentrations where your peptide is linear, and that also is a span of intensities, I guess, and then your intensity or your sensitivity drops in the mass spectrometer like 2 weeks later. Yeah.
Okay, this is your problem. You say that in the moment when you, because, I mean, first of all, you should be able to have a constant running mass spectrometer for a calibration curve. If this is your problem, you need to change something else. Exactly, but if you now say, okay, you have done a calibration curve and now you do measurements in your endogenous samples and you want to use the equation of that curve to quantify. So what's the best way, or what I usually try to do is that you do, of course, not use the absolute intensity that you measure on this one day to generate this curve, but you use a ratio to a reference that you plot, and you use the same ratio to the reference in your endogenous sample later on. So you do. It's of course a bad idea to just compare absolute intensities, but by making them relative to a reference, this can become very easily possible. We can also discuss it later if you didn't get it now. Okay. So absolute quantification. So on this slide, I would like to show you my definition of relative and absolute quantification, and I would like to note right away from the beginning that this is not necessarily the definition of everybody in the field. So maybe there are also people who define this or see this a bit others, but that's my definition, and I will use this for the rest of the presentation. So what I understand about relative quantification is that for relative quantification, you need to work with at least 2 samples. You cannot do relative quantification with just 1 sample. You can, of course, have many more, but you need to have at least 2. And the type of comparisons that you do with relative quantification is that you always compare the same peptide or the same protein between samples. So in these data matrices that we always produce and that we have seen so many times, you always go along the rows. You just focus on the same peptide and make relative comparisons between that. And the output that relative quantitative proteomics gives is then always information about whether a certain protein or peptide changes, what's the fold change that you can see between 2 or more conditions. In absolute quantification, the situation is different. So what I define as absolute is you can also perform absolute quantification with just a single sample. You can do more, but just a single sample is also sufficient. And what you can do with absolute quantification is you can also, of course, go row-wise and always compare absolute concentrations of the same peptides or proteins between samples. But you can also look at different proteins within one sample or between samples. So in other words, with absolute quantification, what you try to do is you try to bring all the proteins onto the same level and normalize all proteins to each other to make comparisons between proteins possible. So the output in absolute quantification can also be a ratio between 2 different proteins in the sample. Or, and that's what I always call the deluxe or the awarded way of doing absolute quantification, is that you also provide a concentration unit to the protein, like that you want to say a certain protein is in a certain number of copies per cell or in nanogram per milliliters of urine present. So to give a real absolute quantification unit. So this is like an extra step that you need to do to get from these relative comparisons of different proteins to an absolute concentration unit. But I call all of this absolute quantification. Well, this is what we're talking about now, how you actually do that. Will come. Okay, so just yeah. So now, if you see this, you might think that it's super cool, or to do absolute quantification, because it gives you much more knowledge. You can suddenly compare a lot more things, and we should all do just absolute quantification. Great, right? Because it's the more informative way of quantification. But there are definitely challenges, and we will discuss these when we now talk about the techniques how to do this absolute quantification. While it's not a good idea to always do absolute quantification, just the opposite. I think you should always, you should just do absolute quantification when it's really necessary for your project to do that. And on that slide, I have summarized some of the typical projects where it does become important to do absolute quantification. And one standard project that really requires absolute data is when you want to determine protein complex stoichiometry. So if you want to look at, in a protein complex, what's the concentration of the different partners towards each other, because to do this, you need to compare different proteins within one sample. Another typical field of application is mathematical modeling. So if you want to determine rate constances, or model kinetic fluxes, or if you want to know how much energy the cell spends on producing certain proteins, you need to know the concentration of these proteins, or the copies per cell of these proteins, in order to solve those equations. Industry is also a typical field of application. Quality control, food safety, sports drug testing, all of these are applications where you need to know the absolute concentration. Protein biomarker development is a field of application. And then there is this data cross-comparison field. So whenever you want to compare data that, let's say, has not been planned to compare right from the beginning, this can be very difficult with relative data, or impossible, because with relative data you always need to have the same reference to compare with, while with absolute data some interesting comparisons might become possible, like between organisms or platforms that were not planned right from the beginning to be compared, because absolute data you can always compare. Okay, so for projects like this, it makes absolutely sense to say, I go this step and do this more challenging absolute quant. But a lot of other projects can be very good addressed with just looking at relative changes, and then you should also query the data in a more relative way. Okay, so now how can we do absolute quantification? So I showed you this figure already before, and if we want to do absolute quantification, what we need to learn, or what we need to know, is this response factor, right? In the moment when we know response factors for our peptides, we can say, okay, this intensity means this concentration of the protein that it had. So response factors is the keyword. And now the question is, how can we determine these response factors? And in the field, there have been label-based and label-free approaches being described that provide this, or that you can use. And definitely the gold standard way is to work with isotope-labeled reference molecules. That's the gold standard way that people in proteomics use to do absolute quantification. And what's very important is that these labeled reference molecules that you work with, you need to know the absolute concentration. And just here, very briefly, once more, the most important facts about these stable isotope labeled references. So stable isotope labeled references carry one or several isotope labeled atoms. Proteomics people love to work with 13C, 15N, 18O. Sometimes also with deuterium. And why these isotope labeled reference molecules are working is because they are chemically identical to the endogenous light targets. And that means they have exactly the same behavior in terms of sample preparation biases. So in the moment when your heavy labeled reference is present in your sample together with your light endogenous target, they will attach to the vessel wall in the same way, and you can account for those sample preparation biases. They will behave exactly the same in terms of chromatography, ionization efficiency. They will fragment the same in your mass spectrometer. And all of these points are the reasons why when you work with isotope labeled references, you can account for run-to-run variations much better than when you work label-free. And if you work with isotope labeled references from which you know the exact concentration, you can learn the MS response factor and do absolute quantification with them. And how do you do this now? To learn this MS response factor, you do something that we discussed already. What we have done already, you do a calibration curve. So you do exactly the same, and that your question was going in that way already. You do what we just discussed when we talked about determining range of linearity. So you spike your synthetic reference in a dilution curve into your sample, and from the intensities that you measure then of your reference, you derive this linear equation, which you can then use to, this linear equation is in a way your MS response factor, which you then use to calculate from the intensity that you measure for your endogenous peptide, the absolute concentration. Okay, so what's very cool about this type of absolute quantification is it's immediately usually the deluxe way of absolute quantification, because it immediately will also provide you a real protein concentration unit. So usually the unit that your isotope label standard was in, which is typically a femtomole per volume unit. And if you want to convert now this unit into other units, like for example, femtomole per microgram of protein extract, you need to know the amount of extracted protein extract that you use to produce your sample. And this people typically determine by BCA or Bradford assay. And we all know BCA and Bradford assay are not so great. So that's the first source of error to convert these units now into each other. For example, also what a lot of people like to do is to report absolute concentrations in copies per cell. However, in order to convert this value and this, you need to know the amount of cells that you use to generate your sample, and you need to know the exact protein extraction efficiency. So you need to know out of the cells that you started working with, how much protein did I extract really 100% of the protein, what people usually assume, or did I maybe just extract 50%? And then the copy per cell number will be affected, yeah?
I think I saw once of these calibration curves that were done in log space. Would that also work, or is this not?
I think.
It makes the low concentration nicer.
Well, so probably Olga would be the perfect person to ask. Even though, so my very basic understanding of that is, if you work in log space, you do not drive your linear correlation by the high abundant peaks, right? Then, I mean, if you do it unlocked, most of the correlation is driven by these high peaks, and it doesn't matter what's going on almost in the low peaks. In log scale, you make like a more normalized view on that. Is Olga here? No, probably not. Huh? Olga, is she still? She's outside. We can go and ask her. Maybe Brandon has also an opinion to that.
Yeah, I've seen a lot. I think that what it's kind of doesn't really allow you to do regression in log space because of the talks with Mike and Andy. Instead, what we would recommend, if you want to give more weight to your small points, then we would suggest using 1/x squared weighting in the regression.
Mm-hmm.
But to always use a linear regression, because the problem is that a log regression is not guaranteed to be linear if the slope is not 1.
Mm-hmm.
And so what will actually happen is you get this curvature sort of toward the lower abundance ones, and so you actually you'll I think overestimate your linear range, because you're curving in the same direction that it's natural for the regression to curve.
Mm-hmm. So, I mean, in the literature, you're absolutely right. I think both things are found and have been done.
I have people who are still asking for log.
Yeah. I mean, that's the.
Yeah. We find we can get that last point in the linear range if we use log. Somebody just told me that.
Yeah.
One thing to remember is when you fit a line in log space, it's no longer a line; it's a power equation.
Mm-hmm.
That's one reason why first thing always.
Mm-hmm. Okay, so yeah, so this protein extraction efficiency is definitely one very weak point and still unsolved point, I think, in absolute quantification in proteomics, because what people do is they assume simply to have extracted all proteins from cells usually. And yeah, there is no tool to really test this or to know this, and so yeah, it's all based on assumptions then. Okay, so these isotope labeled reference molecules, there are several different ones, and again, many different methods that are used. I would say the three main reference molecules people work with in the field are either synthetic peptides or so-called concatenated peptides or completely labeled proteins. And as usual, each of these molecules has specific advantages and disadvantages. So synthetic reference peptides, I guess that's probably what most people work with simply because they are so easy accessible. Typically, people work with C-terminally labeled peptides. So this is the SILAC type of labeling usually. And you can get these peptides quite cheap from various vendors. But the cheap version is always the crude version, so then it wasn't purified. These are fantastic for generating assay libraries, for example, or for reference molecules, but they are not cool for absolute quantification. For absolute quantification, you need to know the exact amount, and then these are these aqua or staple isotope labeled standard peptides. And the major disadvantage of these synthetic peptides for absolute quantification is that you unfortunately cannot spike them into your sample right from the beginning, right? So that's what Olga also already said. Ideally, you would love to have already in the cells, right from the beginning, a labeled reference present. And these synthetic peptides are spiked in after tryptic digestions when you have peptides in your sample. So you cannot account with these peptides for any variabilities that happened before, like protein extraction or digestion variabilities are not accounted for. Concatenated peptides, just very briefly, what is that? A concatenated peptide is an artificial protein where various different, I think up to 50 or 60 peptides of interest are cloned one after another into an artificial new protein. So you create a new protein that just consists out of peptides of interest for you. This can be peptides from the same protein, can be completely unrelated peptides. And then you express this new plasmid, this new artificial protein in labeled medium, either in vitro or in vivo techniques are out there. You purify it, you determine the concentration, and then you spike it into your sample of interest and digest it together with your light proteins, and thereby produce up to 50 peptides of the same concentration, right? They will all have the same concentration if you assume that trypsin cleaves all of them to 100%. Also with concatenated peptides, you cannot account for variabilities in extraction and digestion because you cannot assume that the new artificial protein is digested in exactly the same way as a light protein. But yeah, it can be a really fast way for a lot of peptides in one go that all have the same concentration. And then we have the full-length recombinant proteins. So there you do pretty much exactly the same as the concatenated peptides, but you just use the complete sequence of your target protein, overexpress it, labeled medium, and you spike this into your sample. And there, the big advantage of these reference molecules is that you can account for digestion variabilities if these protein that you produced really resembles your target light protein, which means also in terms of PTMs, which is sometimes maybe again a problem. If you have a PTM on your light protein that is not here, it could still cause issues or miscleavages or something like that. Okay, so overall, no matter with which of these molecules you're working, the big advantage is absolute quantification with isotope labeled reference molecules can be very precise and potentially also very accurate if you have a good working molecule. But the big downside is it's expensive, and that's why people usually don't apply it for full proteomes. And that's when now absolute label-free abundance estimation comes into play. Any more questions to absolute quantification with isotope labeling? Yeah? I mean, this will, of course, very much depend on the reference molecule. So, for example, if you order aquapeptides, it's the company that should make sure it's pure, and they provide you this purity, and you hope it's correct. if you work with recombinant proteins or something like this that you purify, you also, it's the job that needs to be done before, right? You use HPLC or yeah, mass spec data before to determine the purity, and ideally it should be very pure. There's also other approaches where people work with quant tags on those peptides, so that you put a small cleavable peptide to another peptide or to a protein, and you cleave that off, and you use actually this tag to quantify it. So there are many different options depending on with which molecules you're working with. But it's of course a very important feature that should be tested, and it's important. And also one very important, and I just tell it because I was talking also with Amir about it a little bit. What I've also seen sometimes is that these isotope labeled molecules can be impured by the light version. So which means they will not be impure by 50%. The impurity will be 1% or less. But if you then use them as a spike-in and you measure peptide ratios that are bigger than 100-fold, you must be very careful that this could be due to the light impurity that you actually spiked in yourself. So that's one important reason why you should always try to aim for similar peak intensities, light and heavy, then this will not happen to you. But that's just very detailed stuff now already. Okay? Yeah? Mm-hmm. Mm-hmm.
The second question is, in the calibration curve, you take exactly, you take points that they have exactly the difference between the points is exactly the same, like in any bioanalytical chemistry, or you just take many points on the low range and just sum all the big ranges?
So, I mean, you answered, I think, all of your questions already. Yeah, so regarding the calibration curve, it's not super important that they always have the same distance. I don't think so. I mean, what's super important is that you cover the range that is of relevance for you. You need to, and of course, the more points you have, the better, the better you will be able to differentiate that. And but it's very important that, yeah, it makes no sense to make a calibration curve. So what happened to me, for example, I had a calibration curve with the highest concentration of only being 100 femtomole, and it turned out that in my real sample, the analyte was 500 femtomole. It was the most abundant protein in the sample, and it just was very high. So then, of course, it would be very informative to make sure you're not in saturation at that point. So you need to make sure that your calibration curve resembles the range of linearity that you are looking at. How big the distance is, I think doesn't really matter in that sense. By the distance. And what is this? Who demands that? Is it a...
It's, for instance, from the FDA.
Mm-hmm. Okay, yeah, well, there you know more than I know, yeah, so...
I was wondering if also here there is any protocol or guideline
Yeah.
that is telling you that you need to have specific distance?
Yeah, I never heard it. I don't follow then these FDA rules, I'm afraid, yeah, but... I think as long as you cover what you, and that's what I, for example, also often do is when I get aquapeptides, I first make these dilution series, and then I check the intensities in my final data matrix of my real samples, whether the intensities that are in this matrix are covered by the linear dynamic range experiments that I did before. And I would also always provide these linear dynamic range data in a paper, for example, in a supplement or so. Okay. So keep on going. So absolute label-free abundance estimation. So you find in the literature all, or let's say 3 out of the 4 quantification units that I introduced you before, where people have successfully used these units to do absolute label-free quantification. So there are papers that were based on spectral counting that used this approaches, precursor ion intensities measured with shotgun or with MSE data, for example, have been published, and fragment ion intensities measured with SRM and DIA have been published. There has been nothing published for the 4th unit that I showed you, which was the TMT reporter ions. So to my knowledge, I think so far nobody has tried to do absolute label-free with TMT reporter ions. Maybe that would be something cool to do. I don't know. But so far, only those 3 units are reported. And with each of these 3 different units, or with the papers behind these units, there came also different algorithms that were used to absolutely quantify now proteins. And these are just 5 examples that I'm listing here. There are even more. But what all of these different algorithms do is they all try to somehow normalize these differences in MS response factors that I just showed you before. So with all of these algorithms, the aim is always to somehow make sure that we no longer have variability here in these MS response factors, but that we have a constant MS response factors for all the proteins that we're looking at. If you understand this, then you understand the overall approach of all of these different methods. Now, all of these different methods use slightly different equations or mathematics or assumptions to achieve exactly this, and I don't want to go too much in detail, but just, for example, this top-N approach tries to normalize for differences in peptide intensities by saying, okay, I always just focus on the top 3 or top 2 or top 1 peptides per protein. And my assumption is that every protein in a proteome produces 1 peptide that is, or 2 or 3, that is a very good flyer. And they all are so good flyers, they all have the same MS response factor. Okay? This is the basic assumption, and this is how top N works to estimate based on 3 or 2 or 1 top flyer peptide, the absolute abundance of the protein in the sample, because it says the MS response factor of these few peptides is constant over proteome. Now, of course, this holds true for some proteins better than for others, obviously, but over a complete proteome, it has been shown, I think, a few times already, that this assumption holds true surprisingly well for quite a large number of proteins. The IVAC approach is different. So in IVAC, you say, no, I take all the peptides that were measured for a protein. So I sum up intensities of all the peptides that were quantified. And then I normalize this intensity by the theoretical number of peptides the protein can produce. So in a way, by the protein size. So the IBAC approach assumes that the distribution of all peptides, by taking into account all the peptides that are there, the distribution of all of those peptides is something that is constant between all proteins of a proteome. Okay? And then it says, it only depends, the number of peptides that I see, it depends on the size of the protein, so I must correct also for the size of the protein. NSAF and APEX approach are 2 approaches that were originally published for spectral counting data, they can be also applied to chromatography-based data. They're very similar to IBAC in the sense that also in NSAF you sum up all peptide intensities and divide by the protein length, I think. And in APEX, you divide the sum of all peptides by the observability of the peptide. So there is a very complicated smart algorithm behind that predicts for every peptide how likely is it that it was detected or not. And then there is the Scampi approach, which was specifically designed for dealing also with shared peptides, so what to do with peptides that are not just present in one protein. These other approaches, usually they try to avoid this by just focusing on unique peptides. That's what most of the tools, I think, are doing. Okay, so, but no matter whether you use now top N, IBAC, or whatever, the result is always the same. You end up with a list, a quantitative data matrix where you have in each line a protein and then different samples over the different columns. And as this is now absolute label-free, and then intensities, of course. And these intensities were now computed from peptide intensities using one of these algorithms, right? So it's no longer peptide intensity, it's now an inferred protein intensity. And the cool thing is this is now an absolute data is you can now go and compare these intensities, of course, again, relative row-wise over samples, but you can also use these tables and look within one column, within one sample, what's the most abundant, the second abundant protein, and stuff like this. And if you have fancy questions, you can compare different proteins in different samples. Now, this is all this relative data that you then, how you interpret the data, but what about this deluxe way? How can we also now convert these intensities into this real protein quantification unit? And also there, stuff has been published, what people do. A first approach what people use in the field is to use direct proportionality. So it's a very easy approach. All you have to do for that is you need to have a full protein measurement. So you cannot apply this approach if you've just done 10 SRM measurements of 10 proteins, but you need to have covered more or less the whole detectable proteome. And here you see this as an example where I have on this axis the protein count, and this is downranked according to intensity. And now the assumption of this direct proportionality approach is that when you sum up all of those intensities of all the proteins that you have measured, this summed intensity equals the total protein content. Okay, so if the total protein content is 1 microgram, for example, that you load onto the column, and the highest intense protein here makes up 1% of the total intensity, it would be 1% of 1 microgram. Direct proportionality, it would be 10 nanograms, I think, the concentration. And like this, with this, the sum is the total protein content, and knowing the total protein content, you can give microgram concentrations to all of those measured values. So, as I said, to apply this approach, you need to know the total protein content, BCA or Bradford, again, you're bound to that. If you want to determine protein copies per cell, you must know the number of cells and the extraction efficiency. And as BCA, Bradford, and cell counting are prone to a lot of errors, there's also one interesting paper that describes how you can avoid doing BCA or Bradford or cell counting stuff. Called the Proteomic ruler concept, was published by the Mann group. And what they do is they say that the MS intensity of, they look at the MS intensity of histones, so it can only apply to eukaryotes with histones. And they say that the ratio of the intensity of the histones compared to the sum of all intensities is equal to the total protein intensity, or is equal to the amount of DNA to the total protein intensity. So the basic assumption behind this is that there is a constant ratio between histones and DNA in the sample, and this is used to, without doing BCA or Bradford or cell counting, do this total direct proportionality approach. Okay, read the paper in case you didn't understand what I just said. It was not very clear. Okay, and then next to this direct proportionality, there is the calibration curve approach. That's the other general way what people try to do to convert intensities into a real protein value, protein concentration value. And what you do here is you select several anchor point proteins. And again, ideally, these anchor point proteins should span the whole intensity range that you have measured. So from the high intense ones as well as to the very low intense ones. And for those anchor point proteins, you now must somehow know the absolute concentration. So you can, for example, use, again, aquapeptides to measure those, or you can, for example, spike in proteins from which you exactly know the concentration. A good standard a lot of people work with for that purpose is this UPS2 standard. Also, Alexei mentioned already, that's the standard of, I think, 48 human proteins in 6 different concentrations, spanning 5 orders of magnitude. So these can also be the ones when you spike them into a non-human sample. And what you do then is you plot on log-log scale the MS intensity that you measure for those anchor proteins versus their absolute concentration that you know. And from that, you hopefully get a nice linear correlation. And this equation of this linear correlation you use to convert now, again, all the gray intensities into a real protein concentration value. Okay? So, exactly, in case you feel like you ever want to do something like this, get into the field of absolute label-free quantification, I can recommend you to save a lot of work by using a script that was published by George, who will also speak tomorrow, on Friday, I think. And it's called the aLFQ script. It's also freely available on CRAN. And with this script, you can do, or this script does for you a lot of the tests for all those methods that I showed you before with one click. So if you're able to run R, this can save you a lot of time. You can use different quantitative input data sets. You can use spectral counts MS1 intensities or MS2 intensities, you can define the total protein content and do the direct proportionality approach, or you can use the anchor point calibration curve approach and provide this. Then aLFQ will test all these 5 different methods that I just introduced you, and it will give you as an output the calibration curves, and it will do some bootstrapping error estimations, and you can immediately see how on your data set these different approaches perform and which could be a good one for your data. And the last question, of course, is now, how accurate can this be, this absolute label-free? How good will it be? And, or how bad will it be? And there was a nice paper published by Eric Ahne and Alexander Schmidt from the Biozentrum in Basel, who compared these 5 different approaches, again, MPI and mean intensities, again, a bit different, again, other ones, but IBEC, top 3, and APEX I explained you. And they used this UPS2 standard that I also explained you already, spiked that into a bacterial background to have some background. And then they tested calibration curve and direct proportionality approach for all those different approaches with shotgun DDA proteomics. And the 2 best performing approaches they found was IBUC and top 3. And the average error that these 2, the mean fold error that these 2 approaches had was 1.5-fold. So in average, the value that they measured using absolute label-free, top 3 or IBEC, was 1.5-fold off from the true value. And 90% of the proteins had an average error of even below 2-fold. And I think these are numbers that have been consistently published now also by other people and other types of samples. Okay, so is this good? 2-fold, a 1.5-fold average error? Or is this bad? Or what do you think? Good enough? Not so good. Well, so the answer is, as usual, it depends, right? And this brings me back to this figure that I showed you where absolute quantitative data really matters. And I think whenever you have a project where very accurate data really matters, which, for example, are typically protein complex stoichiometry. If you want to determine a 1-to-1 or 1-to-2 stoichiometry, and you have a 1.5-fold average error, this will maybe not be very useful for you. Industry biomarker, highest accuracy is often very, very important. So here I would always say, probably you must go small scale and rather use isotope labeling. But if you have projects where it's important to look at the proteome in a bigger scale, it actually might become very reasonable and very cool to also use absolute label-free quantification, and the bigger error that you have here might be still well tolerated, and you can still draw a lot of meaningful conclusions from such measurements. So it depends. You must make sure it fits to your project. Okay, how much more time do I have? Nothing?
Not much, but take, I guess, 10 more minutes to think about it.
I think it will go rather fast, but we can also stop. I mean, this is the most important. So now I just want to show you one case where we applied this.
I think you can.
Okay, good. So, so that's a project that was led by Olga Schubert, a former PhD student in the lab. And her PhD thesis was about studying virulence and resistance in Mycobacterium tuberculosis. MTB, as you probably all know, is the causative agent of tuberculosis. 1/3 of the world's population is latently infected, many deaths every year. And the problem with MTB is that it has an extraordinary ability to survive under conditions that are very bad for the organism. And it achieves this by entering what's called a state of dormancy. So it's going to sleep. And in this sleep state, you can hardly kill it with antibiotics, and it just stays in your body and it can get active again. So the goal of Olga's thesis or of this project was to gain a better understanding of the mechanism underlying this dormant state. So what's happening there, and how can you maybe find ways to still kill MTB even when it's dormant? Okay, and the experiment that Olga did was to grow MTB in shake flasks and to decrease the oxygen level in this shake flask, because due to lack of oxygen, hypoxia, MTB goes into exactly this dormant state. And she simply decreased the oxygen by closing the lids. So the oxygen was slowly consumed. And she did that over 20 days. And after 20 days, she opened the flasks again and looked at how the cells started regrowing. So in total, there were 6 time points taken in biological replicates. Then we extracted the proteins, digested them with trypsin, and after trypsinization, we added 60 aquapeptides for 30 proteins, which we wanted to use as anchor proteins to perform this calibration curve conversion of intensities into absolute quantitative data with a real unit. All the samples were then measured by SWATH, and they were analyzed using an MTB SWATH assay library that contains assays for 98% of the MTB proteins, so that's a super nice and high number. And this library was generated before from Olga, and this number is so high because she used over 17,000 synthetic peptides for MTB that were synthesized and that were predicted to be good peptides for proteins which had never been seen before by mass spectrometry from this organism. She also used strongly fractionated lysates, and she used several different growth conditions, and all of that went into this final library, which is now also available on SWATH. So if you work with MTB, that's maybe a very cool resource for you. Yeah, so this library was used to then analyze the DIA SWATH data. And we used OpenSWATH at that time for that, and we used this aLFQ script that I showed you to look at all the different methods. And the best method on that data set that we found was the top 3, so the best 3 peptides quantified with, I think, 5 or 6 fragment ions each, was the method that, according to aLFQ, gave us the best accuracy for absolute quantification based on these 30 anchor proteins. Exactly. So, and now here you see how the data looks like. So this is this table, typical quantitative data metrics. We got for over 2,000 proteins, we got this absolute label-free quantification estimates without missing values. And what you can do now with this data set, as I told you, you can start looking at one column, and you can look what's the most abundant and the least abundant protein in this, and you can look at the dynamic quantification range. So the highest abundant protein was almost 100 copies per cell, protein copies per cell. The lowest one was in the range of 10 protein copies per cell that we could measure. Another analysis that you can do on this type of data set is you can look at how different functionally related proteins contribute to the total proteome biomass. So this is what you show here. So these were different functional annotations of the MTB proteome. And under normal growth conditions, you can see that, for example, the lipid metabolism makes up 10% of the biomass that is present in the cell in terms of proteins. A ribosome makes up maybe here 7%. And we found interestingly that when now the bacteria go into dormancy over these time points, this general contribution to the biomass did not change a lot for all classes except for one, and that's this was a stress rigolon containing 50 stress proteins, and yeah, this went up to a number of almost making up 20% of the total protein content in the cell, and that's this type of information that you can just get from absolute and not from relative data. And one final last example that I would like to show you is the example of mathematical modeling. So we also used the data for some modeling. So we looked here into kinetic fluxes, flux changes, and one interesting tiny result that came out, which I think nicely visualizes this additional benefit of absolute data, concerned here these phosphofructokinase A and B isoforms. So these are 2 proteins, 2 enzymes that catalyze the conversion of fructose 6-phosphate to B-phosphate. And if we would have just used our data in a relative quantitative way, the situation looked like this. You can see now these are now relative fold changes, and in our data set, the protein PFKA was not at all changed during dormancy compared to normal growth, while the isoform PFKB went up like crazy, 6-fold, was much higher. So the logic conclusion from such relative data would, of course, be PFKB is a super important protein during dormancy. Now, that was what the relative data looked like, and when we converted now, when we used the absolute label-free quantification and did absolute quantification, what we could do is we estimated the Vmax, that's the enzyme capacity or the maximal reaction rate that these two isoforms can have by simply multiplying the turnover frequency with the absolute concentration. And when you look here, the situation now was completely different. So the enzyme capacity, the maximal reaction rate, was still much, much higher for PFKA than it was for PFKB. PFKB goes higher, yes, but it's still way below PFKA. And this is just showing you, so the conclusion from this figure is that PFKA is also in dormant state, the major protein contributing to the glycolytic flux for this pathway. And yeah, I think that is a nice example that highlights that this absolute quantitative data is really, really more information rich. And actually that this is the true scenario was also evidenced by PFK mutant, that PFK deletion mutant that could not, where PFKB could not functionally complement this mutant. So it's shown that this scenario is really rather the true one. Okay, with this I'm at my take-home messages. Are there any more questions to MTB and absolute label-free quant? It's also very late, I'm sorry. So take-home messages, what I think is most important to keep in mind is protein quantification. Everything we do, no matter with which technology is always based on the assumption that MS intensity scales linear with protein abundance. And this linear dynamic range is peptide sample and mass spectrometer dependent. There are many different ways how you can do quantitative proteomics. They can be categorized in relative and absolute. And my quantification of relative is you always compare the same protein, the same peptide between samples. In absolute quantification, you can look at the whole matrix because the proteins are all normalized or brought onto the same scale. And yeah, that's why absolute quantification can still be relative also in the sense that you just look at ratios between one sample, or you can do the deluxe way of also determining an absolute concentration. Absolute quantification is challenging, especially if you want to be accurate, and it's expensive, and you should just apply it to specific projects. And the major advantages of doing label-based quantification is you're more precise and you're possibly more accurate if you have a good reference molecule, but it's expensive and just applicable to small scale. Label-free can be protein-wide, but keep in mind the error can be in average around twofold, which means the average is twofold. There can be specific proteins that have a much bigger error, right? And you just don't know which are the ones with a significantly bigger error or not. With this, I would like to say thank you for your attention. Thanks a lot for the organizers for having me here again, and to my team in Munich. Thanks, and I'm around until Friday, so you can come anytime and ask me questions.
Great, thanks a lot.